everyone, welcome back to my class. This is Rumana Ali. I hope you all are doing well. In the last session, I have explained you about the different digestive glands like salivary glands, intestinal glands, gastric glands, and even we have learned about the largest exocrine gland, that is our liver. Now in today's class, let us learn about the functions of liver. So the functions of liver includes the metabolism of carbohydrates, metabolism of fats, and even the metabolism of proteins can be achieved by the organ, that is liver. So if the metabolism of all these nutrients would not be getting in a proper manner in our body, then many different problems can arise. So metabolism is very important. Without this, many organs would get affected. So liver is playing a major role in metabolizing carbohydrates, fats and proteins. Now let us see how the metabolism of carbohydrates takes place. So glucose is a form of sugar. This glucose is broken down into the glycogen in the liver itself. So liver is playing an important role in breaking down the glucose into a new form, that is in glycogen, and this process is called as glucogenesis. Enough amount of glucose is not present in our body. At the time, this glycogen can be converted back into glucose, and this process is called as glycogenolysis. So both the process, glucogenesis and glycogenesis, takes place in the liver, and this is how the metabolism of carbohydrate takes place. Now the proteins and amino acids or the fats which are present, it can be broken down in the liver itself, and that process is called as gluconeogenesis. So this proteins, amino acids and fats, they can be broken down into a form of sugar that can be consumed by our body, and that sugar is nothing but the glucose. So this is how the metabolism of carbohydrates takes place by the liver. Now let us see how the fats are going to break down. So fatty acids, fats are present in the food item. In the different food products, we can see the presence of fats in the form of fatty acids and glycerol. So this fatty acids and glycerol, they are going to converted in the form of a fat. So this fat can be converted back into fatty acids and glycerol. This interconversion takes place in the liver only, and when the fatty acid and glycerol is broken down into fats, then that process is called as lipogenesis, and when the fat is converted back into fatty acids and glycerol, that process is called as lipolysis. So lysis means breakdown. When the fats broken down and converted back into the fatty acids and glycerol, then that process is called as lipolysis. So all the reactions like glucogenesis, glycogenolysis, gluconeogenesis, lipogenesis and lipolysis, all these kinds of catalytic reactions takes place in the liver. And liver also helps in the metabolism of proteins. So during the metabolism of proteins, there are two main reactions that takes place. The first one is transamination and the second one is deamination. Transamination means transfer of one amino group from one amino acid to the other. So amino acids contains amino group. When this amino group is transferred to the other amino acid, then it is called as transamination. This is how the amino acid would get synthesized, and when this amino acids are joining together, they form a chain-like structure, and that is nothing but the protein. Next one is deamination. Deamination means dissociation of the chain of amino acid. When the amino acid chain is going to break down to release some compounds, then that process is called as deamination. The other function of liver is it helps in the conversion of toxic. So other function of liver is it helps in the conversion of toxic amino group into less toxic urea. So amino group or ammonia is very toxic in nature, and that can be converted in the liver. So we can say that liver is an organ that helps in the process of detoxification. Toxic substances can be converted into less toxic one. This process is called as detoxification. The next one is liver is an organ that plays an important role in the process called erythropoiesis. Erythropoiesis is a process where the formation of RBC takes place. Red blood cells. Production of red blood cells can occur in the liver, that too only in the embryonic life. This point should be mentioned. Whenever we talk about the process of erythropoiesis that takes place in the liver, we should mention this one, embryonic life, because only in the embryonic life, during embryonic stages only, liver plays an important role in the formation of red blood cells. But when the baby born, at the time, the red blood cell production would occur in the bone marrow of our body. These are some of the functions of an organ called liver. The next function of the liver is in thermoregulation. Thermo means heat in our body. The distribution of heat is also an important role that is played by the liver. So it also helps in the process called thermoregulation. Liver is an organ that stores fat soluble vitamins. Fat soluble vitamins are nothing but A, D, E, and K. These four are termed as fat soluble vitamins which can be stored in the liver. And liver is a place where the plasma proteins, different plasma proteins can get synthesized. For example, albumin. Albumin is a plasma protein that can be synthesized in the liver, and besides this, some clotting factors can also be synthesized. It can also be produced in the liver, and that clotting factors are fibrinogen and prothrombin. These two clotting factors plays an important role in the process blood clotting. These clotting factors can also be produced. It can also get synthesized in the liver. And liver is an organ that helps in the process called detoxification. Just before, I already explained you that it helps in the conversion of more toxic substances into the lesser toxic one. So this process is called as detoxification, and it takes place in the liver. Liver is an organ that plays a key role in the production of bile. We all know that bile is a juice that is composed of some acids, salts, and some other pigments. So bile is a juice that helps in the process of digestion, and this bile can be produced in the liver. The liver can produce this bile. Bile is basic in nature because its pH is 8. Its pH is approximately 8. So this bile contains bile acids, bile salts, bile pigments. Bile acids are cholic acid, deoxycholic acid, and chenodeoxycholic acid. These kind of acids helps in the conversion of starch, helps in the conversion of complex substances into the simpler one. And bile salts are also present like sodium bicarbonate, sodium glycocholate, and sodium taurocholate. These kind of bile salts helps in the process of emulsification. So what do you mean by the process emulsification? It's a process by which the fats can be broken down. The breaking down of fats is called as emulsification. Bile pigments like bilirubin and biliverdin are also present, and these pigments are yellow in color. So the fecal matter which comes out of our body, that is yellow in color due to the presence of these two pigments. These bile pigments are present in the fecal matter that gives yellow color to it. So this is how the liver is playing many different roles that are very essential for our body to work properly. If the functions of the liver gets affected, then the whole body would get affected. So these are some key functions of the largest exocrine gland that is present in our body is liver. So up till we were discussing about an exocrine gland that is liver. Now I am going to explain you about another gland which plays an important role in the process of digestion, and that is our pancreas. So we'll discuss about the structure and functions of pancreas. Firstly, we'll see where it is located. So it is located just beneath the stomach, and it shows 4 main parts, and that parts are head, neck, body and tail. When you look at the diagram, you can easily recognize what are the different parts that are present in the pancreas. So this part, which is located just below the sphincter of the stomach, that is the head, and here one constriction you can see, and this part is called as neck, and this is the body, and the last part of the pancreas is tail. So in this way 4 parts can be seen in the pancreas, and pancreas is called as heterocrine gland. Why do we call pancreas as heterocrine gland? So this is named because of its function. Based on its function, we call the pancreas as heterocrine gland, because the pancreas is an organ that acts as both exocrine gland and endocrine gland. It is showing both the parts, exocrine part and endocrine part, in its structure. So exocrine part, which is present in the pancreas, it shows the presence of some specialized cells, and that cells are called as achinal cells. These cells are responsible to secrete pancreatic juice, and this pancreatic juice can be transferred for the process of digestion through the different ducts that we have already discussed in the last sessions. Now endocrine part shows the presence of some specialized cells again, and the cells which are present in the endocrine part of the pancreas, they are islet of Langerhans, and these islet of Langerhans plays an important role in the secretion of different hormones, and they help in the secretion of one important hormone, that is insulin. So in this way pancreatic juice can also be secreted, and different hormones can also be secreted by the pancreas. We can call the pancreas as heterocrine gland. Now let us look at the structure of exocrine part and endocrine part of the pancreas. So when you see the exocrine part of the pancreas, it shows the presence of some duct. So whenever the duct is present, that gland is called as exocrine gland, and the gland which is present as the ductless gland, that can be termed as endocrine gland. So this is the exocrine part of the pancreas, where you can see the cells called achinal cells. In this way the achinal cells are arranged, and some ducts are coming out of these achinal cells. So whatever the pancreatic juice is that is secreted by these cells, it can be collected by the different ducts, and one common duct is present through which this pancreatic juice can be transferred. So this is how the achinal cells are present that helps in the production of pancreatic juice, and that pancreatic juice can be transferred for the process of digestion through a duct that is called as pancreatic duct, or we can say duct of Wirsung. The duct which is present in the pancreas, that duct is called as duct of Wirsung. Next one is endocrine part, or when you look at the endocrine part of the pancreas, then you can see the presence of different specialized cells. Mainly 4 different kinds of cells are present in the endocrine part of the pancreas, and you cannot find any duct here. The secretions that are going to be secreted by these cells, they will be mixing up with the blood directly without passing any duct. Without any duct, they are going to be mixed up with the blood. So that type of secretions are going to be secreted by these different cells. So what are the different cells that you can find in the endocrine part of the pancreas? The first one is A cells, which are present at the peripheral portion. A cells are also called as alpha cells, and these cells plays an important role in the secretion of an hormone called glucagon. So glucagon is an hormone that is secreted by alpha or A cells of the pancreas. And the next kind of cells that are present in the pancreas are B cells, or they are also be called as beta cells. So B cells or beta cells are also found in the endocrine part of the pancreas that helps in secreting a hormone called insulin. The next one is FF, or we can call it as P cells. So FF or P cells are responsible to secrete pancreatic polypeptides, and the last one is D cells. D cells are also called as delta cells. These delta cells plays an important role. These delta cells helps in the secretion of a hormone called somatotrophin. So alpha or A cell secretes glucagon, beta or B cell secretes insulin, FF or P cell secretes pancreatic polypeptides, and D cells or delta cells secrete somatotrophin. In this way, different hormones are going to be secreted by the different cells that are found in the pancreas. So pancreas is also an important gland that acts as both exocrine gland and endocrine gland. Exocrine gland means how it acts as exocrine gland? Because it helps in the secretion of pancreatic juice, and that pancreatic juice can be transferred through a duct, helps in the process of digestion. And how it is acting as an endocrine gland? So the different cells are present in the pancreas that helps in the secretion of hormones. So on the basis of this function, it can be termed as an endocrine gland. So this is about the structure and function of pancreas. Now let us study about the digestion of different nutrients. So first one that we are going to learn is about the carbohydrate digestion, how the digestion of carbohydrate takes place in the different parts of the alimentary canal. So firstly, the food that we eat, it contains different disaccharides and polysaccharides in it. Disaccharides like sucrose and lactose are present, and polysaccharides like starch, glycogen and cellulose, all these kinds of different saccharides are found in the food that needs to be broken down in order to release energy. So after chewing the food, the mechanical digestion would take place, and after the mechanical digestion, the food that we eat, it can be called as the bolus. Bolus is nothing but the soft food. After chewing the food that becomes soft, it can be termed as the bolus. When the bolus reaches the stomach, so after a little bit of digestion, that partially digested acidic food is termed as chyme. Now let us see how the carbohydrate digestion can be done, how it takes place in the different parts. So firstly, we'll look at carbohydrate digestion in the buccal cavity. We all know that in the buccal cavity, saliva is present that secretes an enzyme called salivary amylase. So salivary amylase is an enzyme that plays a key role, that plays a crucial role in the digestion of carbohydrates. So starch is a polysaccharide that can be converted into simple one like maltose, isomaltose plus limit dextrin. This starch is digested by an enzyme called salivary amylase, and only 30% of the starch can be digested by the action of an enzyme called salivary amylase that takes place in the buccal cavity. 3% of digestion can be done in the buccal cavity, and remaining 27% of digestion is going to take place in the stomach by the action of an enzyme called salivary amylase. Now let us move to the next part, that is in the stomach. After the proper digestion of starch that is present in the food by the action of salivary amylase, when it reaches the stomach, at that time here in the stomach, no carbohydrate digestion enzymes are present in the gastric juices. We know that the stomach is a part that secretes different juices like gastric juices that helps in the process of digestion, but this gastric juice do not contain any enzyme which helps in the digestion of carbohydrate. So as there are no enzymes, no amylases are present in the gastric juice, no carbohydrate digestion takes place in the stomach. Next part in the alimentary canal that helps in the carbohydrate digestion is our duodenum. Duodenum is the first part of the small intestine. So the stomach opens into the duodenum. When the starch reaches the duodenum, at that time some enzymes are present, and that enzymes are pancreatic amylase. Pancreatic juice is secreted by the pancreas that is transferred to the other parts like to the duodenum. So in the duodenum, pancreatic amylases are present which acts on the starch, and these amylases are able to convert 70% of the starch into the different sugars like maltose, isomaltose and limit dextrins. These maltose, isomaltose and limit dextrins are also the complex one. These cannot be called as the simple sugars. So these substances are also be undergoing a reaction to convert themselves into the simple one. So let us see what are the different enzymes that are responsible to convert these sugars into the simpler one. So first one is maltose. Maltose, in the presence of an enzyme maltase, can be converted into glucose. And sucrose, in the presence of an enzyme sucrase, can be converted into glucose plus fructose. Glucose, fructose, these are the simple sugars. And after the breakdown of this complex into the simple sugars, we can get the energy from that simple sugars. Lactose is a compound that can be broken down by the action of an enzyme lactase into glucose and galactose. And isomaltose, in the presence of an enzyme isomaltase, can be converted into glucose and glucose again. And limit dextrin is also a compound that can be converted into glucose by the action of an enzyme limit dextrinase. So all these enzymes are present in the duodenum, which acts on the different sugars and convert them, break them into the simple sugars that can be absorbed easily by our body. So this is how the carbohydrate digestion takes place in the buccal cavity and also in the duodenum of our body. So that's all for today. Meet you next.